Okay, this is the new key to the group problems. This is the COVID edition. These are the changes that I had to make for the loss of the one day because of the school holiday in terms of materials. And so here is just a little review, much like we would have done with group problems for lecture exam 2. So the first question is the same as it was originally, and basically asked to outline the major steps for the complete oxidation of one glucose molecule. And I want you to include the ATP, NADH, FADH2, CO2, and things like that. Assume that the total amount of ATP is 40 in this case. Knowing that it's 40, it tells us a couple things. 40 total ATP is the same as 38 net ATP. Which means it's a passive transport mechanism that is used to shuttle the NADH that we make in glycolysis in the cytosol into the mitochondria in order to convert it into ATP. The steps we need to do that would be 4 sequential steps: glycolysis, activation of pyruvate, the Krebs cycle, and then the electron transport chain. Remember, the electron transport chain, or also known as oxidative phosphorylation, is not accounted for in terms of the summary tables we'll look at and things like that, just because we double counted if that were the case. So looking at the schematic, we'll just look at the top part first. What we do is we take glucose, which is a 6-carbon sugar, add 2 ATPs, not that you have to know this, but into glucose 6-phosphate and then glucose 1,6-bisphosphate. That splits into 2 separate molecules, which eventually become 2 3-carbon pyruvates. And along that way, when we split up, we make an NADH for each pyruvate, and then 2 ATPs for each pyruvate. One of the problems we have is that the NADH that we make in glycolysis, again, it's in the cytosol, and that has to be able to get into the mitochondria to make energy. Mitochondria is a membrane-bound organelle, which means that only small nonpolar molecules can pass through it directly. So therefore, the NADH, being a large nucleotide, it's got sugar, it's got phosphate, it's got a nitrogenous base, it's got some energy on it. It's a very polar molecule. It can't get through. So what it does is it passes its energy to malate, which then brings it across and then passes it back to NADH inside the mitochondria, and eventually aspartate comes back out, and it just keeps on going back around to bring NADH into the mitochondria, which we can then cash in for energy at a later step. The 2 ATP we don't have to worry much about for each one that we make. And then the pyruvates, the fate of pyruvate depends on the condition of the cell. And anaerobic situations, it'll be converted into lactate. In aerobic situations, it will diffuse into the mitochondria and become activated, becoming acetyl coenzyme A, and then making energy from that. So the second step is activation of pyruvate. Once the pyruvate has entered the mitochondria, we basically cut off the CO2 molecule, so it goes from a 3-carbon pyruvate to a 2-carbon acetyl coenzyme A. In that process, we get 1 NADH, and that carbon comes off as CO2. For each activation step, from turning pyruvate into acetyl coenzyme A, we get 1 NADH and 1 CO2. However, for a glucose molecule, since we get 2 pyruvates per glucose, we get 2 of these steps happening. And so we also, on the other side over here, yield an NADH and a CO2 as well. Once the activation step is over, then the acetyl coenzyme A that we made, that 2-carbon molecule, is free to go into the Krebs cycle. What it'll do is it'll mix with a molecule what's called oxaloacetate. Oxaloacetate is a 4-carbon molecule. Acetyl coenzyme A has 2 carbons. So what's going to happen is they're going to form together to make citric acid. Hence the name of the Krebs cycle sometimes is also known as the citric acid cycle. Occasionally the tricarboxylic acid cycle because we make those during the process, or just TCA for short. So citric acid is the first step that you do in terms of the citric acid cycle. Once we got citric acid, the citric acid goes through a number of steps, and we're not going to worry about those individual steps, but basically the summary for what's called one turn of the Krebs cycle, where we took one acetyl coenzyme A and put it in, we get 1 ATP. Remember, that's direct. ATP is direct, or also known as substrate level phosphorylation. We get 3 NADHs, 1 FADH2, and since we went from a 6-carbon to a 4-carbon, we lost 2 CO2s. So that's the yield from one turn of the Krebs cycle. Since we made 2 pyruvate molecules, then we'll have 2 turns of the Krebs cycle for this. Sometimes an easier way to present the data is to look at it in a table. So here's a summary for each of the 3 major steps. Again, the electron transport chain, where it's understood it's happening, we don't have to count the energy because we've already counted for it in a slightly different way here in the table. So if we take glycolysis, we use 2 ATP to start. Notice it's the only place in metabolism that we're going to talk about here that uses ATP. We get 4 ATP direct. That means we get 2 ATP per pyruvate molecule, right? That again is substrate level phosphorylation. Also glycolysis having the cytosol. We get 2 NADH net, and even though they are made outside of the mitochondria, when they get shuttled in by the malate aspartate shuttle, they end up inside the mitochondria as NADH as well, so we don't have to really change their fundamental nature. We don't have any FADH2s here. My indirect ATP comes from how many ATP do I get from NADH and how many do I get from FADH2. Every NADH molecule is multiplied by 3, so that gives me 6 ATP. Every FADH2 is times 2, so 2 times 0 is 0. So 6 plus 0 is my indirect ATP. That's how much energy I get by NADH and FADH2. You could also say oxidative phosphorylation, or even the electron transport chain would all relate to our indirect ATP. So the energy equivalents for FADH2 are 2, so we get 2 ATP per FADH2 molecule. The energy equivalents for NADH are 3, so we get 3 ATP per NADH molecule. So we just take how many we get from NADH and how many we get from FADH2, and that's my indirect ATP. To get total, I take the direct, the indirect, add them together, so that's 10. To get net, I take the 10, subtract 2 used at the beginning, and that gives me 8. Since we have 2 3-carbon molecules, and we started with the 6-carbon glucose, we've conserved the carbons, no CO2 has come off. Activation and Krebs both occur within the mitochondria. The activation step produces 2 NADHs. We go from, again, that 3-carbon pyruvate to a 2-carbon acetyl coenzyme A, and in that process, giving off a CO2 for each step, which gives us 2 total CO2 and 2 NADH for an entire molecule. So since we have 2 NADH, we multiply by 3. We still don't have to worry about FADH2 here. So 3 times 2 is 6 from our indirect. My total is direct and indirect, so 0 plus 6 is 6. My net is total minus used, so 6 minus 0 is 6 still. And we do have to account for the 2 CO2s that came off our pyruvate molecule to form acetyl coenzyme A. Once we have the acetyl coenzyme A through the activation step, now I just combine with a 4-carbon oxaloacetate, or what is often known as OAA. We then go through a series of reactions that are referred to as the Krebs cycle, and we get the energy yield here. So we get 2 direct ATP through substrate level phosphorylation. We get 6 NADH net, 3 on one turn of the Krebs cycle, 3 on the other, so that's 6. We get 2 FADH2s, that means there's 1 net FADH2 on one side, 1 net FADH2 on the other. So to kind of do the energy summary, we've got 6 NADHs times 3 is 18, 2 times 2 energy equivalents for FADH2, so that's 4. So 18 NADH, 4 FADH2 gives me a total of 22. Total is indirect plus direct, so 2 plus 22 is 24. Net is total minus used, 24 minus 0 is still 24. My CO2 net, remember we went from a 4-carbon molecule to a 6-carbon originally, and then those 6 carbons came off as we went through a metabolic pathway. Since those came off as CO2, we count them there. Looking at the summary, the key component for us is the 40 total ATP, since that's what the question asks for. Net would be 38 when we take that 2 used. The ATP indirect is 34. The way you can do that is 6 plus 6 is 12, plus 22 is 34. That's one way to do it, and that's probably the easiest. The FADH2 net, in this case, we only have 2, which gives us 2 FADH2s, which doesn't give us very many ATPs, but that would give us 4 ATPs total. The NADH is 2, 2, and 6. Remember, we make 3 NADH per turn of the Krebs cycle, and since we have 2 turns of the Krebs cycle, then we have a total of 6 NADHs here. In the activation step, we got 2 NADHs, and then the NADHs we made in the cytosol during glycolysis stayed NADHs because we passively transported them in by the glycerol phosphate shuttle. So we have a total of 10. So 10 times 3 is 30. 2 times 2 is 4. So they're 34. Direct ATP is 4 in glycolysis, 2 in the Krebs, so that's 6. 6 plus 34 is 40. So that's kind of a nice energy summary. All of this has some information to help you understand what's going on and kind of go through the various steps. But here it is for that. So that was question 1. Question 2 basically says when we break down glucose through glycolysis and we get 2 NADH molecules, the fate of those NADH molecules is dependent upon the condition of the cell and the type of cell where they're made. Please discuss the various fates of these NADH molecules and how much energy they are worth. So, during glycolysis, right, we make 2 NADHs, 1 for each pyruvate we make for that split. So we have those 2 NADHs, and the question is, what's the fate of those NADHs based on the various cells and conditions within those cells? So the first place we're going to look at would be anaerobic glycolysis. So during anaerobic glycolysis, which is also known as fermentation, the pyruvate that we produce at the end of glycolysis, by definition, needs to be converted to lactate. For anaerobic glycolysis, the end product is always going to be lactate in humans. So the anaerobic glycolysis gives us no energy from that NADH, and here's the reason why. In order to make lactate, we have to take the NADH that we made in making pyruvate initially and use it up to convert pyruvate into lactate. And so we're going to use up that NADH and make it NAD plus again. That NAD plus then allows high metabolic processes to occur Even though NADH is worth 3 ATP in the mitochondria, for anaerobic glycolysis, since we don't have oxygen, we can't use the mitochondria, so there's no immediate energy yield from that. Yeah, it's the energy stored in lactate indirectly, and eventually we could, make more glucose through gluconeogenesis or something. We're going to have a loss of energy there, and a net loss overall. But for the most part, that's anaerobic glycolysis. Pretty simple. We take the NADH and convert it to NAD+, so we can continue to provide NAD pluses for the anaerobic glycolysis to take place and keep on moving through glycolysis. The second way we can look at that is aerobic respiration in the liver, and that includes most cells. In liver cells, the 2 NADH molecules we make in glycolysis, which again are in the cytosol, they produce 2 pyruvate molecules. But this time, since we have oxygen, we can shuttle these into the mitochondria to make ATP. In the case of the liver anaerobic respiration, we would use what's called the malate aspartate shuttle, a process that allows us to passively move NADH into the mitochondria. There's no expenditure of energy, and then within the mitochondria we can convert the NADH ultimately back into ATP. Since there are 2 NADH molecules per glucose molecule, since there's 2 pyruvates, we can kind of go ahead and calculate the energy yield again. Since 3 ATP are made from 1 NADH, right, our energy yield would be 6 because we have 2 NADH made during that process. The other thing we could look at here would be what happens in skeletal muscle. And skeletal muscle is fundamentally different in many ways than, say, the liver. But in skeletal muscle, the process continues basically the same way. And at the end of glycolysis, we have 2 pyruvates that have to diffuse into the mitochondria somehow and make energy. And we also have 2 NADH molecules that we use. And to go ahead and figure out what we need to do with those. So the NADH cannot be converted into energy when it's outside the mitochondria, and glycolysis happens in the cytosol, which is outside the mitochondria. So these have to be shuttled in via what we often refer to as the glycerol phosphate shuttle, and that actively moves NADH into the mitochondria, where it can eventually be converted to ATP. Since this is an active transport process, there's less energy available at the other end, and since there's less energy available, once it is in the mitochondria, it works. But when so we use the glycerol phosphate shuttle to move the NADH across. This is an active transport mechanism where we can move NADH against its gradient into the mitochondria if we need to, and we don't have to wait for a energy so we don't have to wait for an energy gradient into the cell. We can actively transport that in. The problem is that requires energy, and by the time we get into the mitochondria, the energy we had from NADH is now only available to pass to an FADH2 molecule. These are then used in electron transport to make 2 ATP each, which gives us 4 ATP total from those 2 NADH molecules produced in glycolysis. So as a quick summary, the anaerobic glycolysis, because we don't have any NADH anymore, we use it to make NAD+ and lactate, and that allows glycolysis to continue. That gives me 0 ATP for those 2. In aerobic respiration in liver, The NADH we make is passively transported through the malate aspartate shuttle into the mitochondria. Each one gives an NADH in the mitochondria as well. So that's, since it's 3 ATP each, that gives me 6 ATP for the 2 NADHs. In skeletal muscle, it's actively transported across the membrane by the glycerol phosphate shuttle. It costs a little energy, but now we can move energy in at a faster rate, even if it's against an energy gradient. This is especially useful when you have high metabolic needs. By the time we end up in the mitochondria, we can only pass the energy to FADH2 molecules, since these are worth 2 ATP each, and we have 2 FADH2 molecules. Our energy yield is 4 ATP for those 2 NADHs in skeletal muscle. Next question looks at Cannon's 4 postulates in regards to homeostasis. Cannon was very influential in the field of physiology, and especially homeostasis. As an example, he gave us the fight or flight terminology, but this is another important contribution. He published these, officially in 1929, but worked on them significantly earlier. Doesn't really matter the order they're in, but the first postulate is the autonomic nervous system plays an important role in this process. So Cannon believed that the ANS played a significant role in how homeostasis worked, and he believed that it worked primarily below the level of consciousness. It was mediated primarily through the ANS then, and research has shown subsequently that through especially the effects of the sympathetic and parasympathetic nervous systems, they're essential in terms of maintaining various variables in our body. For instance, minute-to-minute changes in blood pressure controlled primarily through changes in ANS, whether it's, as an example from we learned today, in other areas, a alpha-1 receptor having a norepinephrine molecule bind to it in a systemic blood vessel and constrict it and increase blood pressure, as an example, would help regulate that. And there are so many other factors from the ANS that lead to changes in not just heart rate or stroke volume, or what I just explained a second ago, what's called total peripheral resistance. So the ANS does certainly play an important role. Number 2, there's always some level of tonic activity. Number 2 indicates that there's always some level of activity going on, and in that activity, there's some level of whatever's happening, whether it's tonic control for a nerve or some level of hormone. Humans all have the same hormones unless there's some sort of decrement in your physiology. This means that men and women both have, testosterone, estrogen, human chorionic gonadotropin, which is used to assess pregnancy on a pregnancy test you buy in the store. But the tonic activity tells us there's always some level, but that level may not be high enough to have an effect. You see the same thing with stimulation of neurons to various parts of the brain. There's always some sort of tonic activity. And that allows us, especially if there's only one branch that goes there, if you only have mono-innervation instead of the dual innervation, then having tonic control allows you to be able to change those levels a little more. The next postulate that Cannon came up with was the idea of antagonistic controls. We've seen this in numerous places in the body, where having the ability to move up or back down to a set point and maintain homeostasis is essential. There are a number of different examples, but if body temperature went up, you would have mechanisms to cool you down. If it went down, you would have mechanisms to bring you back. And so those are antagonistic control. Another common one would be the use of glucagon and insulin. Insulin decreases blood sugar. Glucagon antagonistically increases blood sugar. So there's many examples of antagonistic control. The fourth postulate would be it's the receptor that gives the effect, not the chemical. So what it means is we could have the exact same chemical in our body, and if it binds to a different type of receptor, it could have completely the opposite effects. A great example of that is norepinephrine. When norepinephrine binds to an alpha-1 receptor, it causes smooth muscle to contract, and it constricts a blood vessel, as an example. The same exact chemical, norepinephrine, also can bind to a beta-2 receptor. A beta-2 receptor, when it binds norepinephrine, actually causes smooth muscle relaxation or vasodilation. And so, even though we have the exact same chemical, in one part of the body it causes constriction, and the other part of the body it causes dilation. So that's how these different postulates for homeostasis work.